And our second MOOC speaker for today is Antonio Di Guardo. He is a professor at the Department of Science and High Technology at the University of Insubria. Much of his work focuses on fate transport and exposure modeling of chemicals in the environment. He is the invited expert of the exposure work package. And today he'll be giving a MOOC on dynamic models for the prediction of fate of chemicals in the multimedia environment. So Antonio, thank you very much, and the floor is now yours.
Thank you very much, John, and thank you, the organizer, all the people, for inviting me to give this presentation. This presentation will be mostly based on our experience as environmental modeling group at the University of Insubria, and of course the collaboration in the project we've been going through. And I thought this could have been the best way to interact with the people and the PhD student in the group. So I will start thanking the collaborators, my past collaborators, and Melissa Morselli, Davide Ghirardello, Alfonso Infantino, and the current collaborators, Elisa Terzaghi and Parisa Falaktin, who is a PhD student. And start with a very short outline, just to give you a few ideas. So really a few words about environmental fate models. What are we talking about? Of course, fate models are many of different types. And I will go into three main sections, a little bit of terrestrial, although most of the attention in this workshop is on aquatic models and aquatic aspects. So we'll talk a little bit about terrestrial models, but also aquatic dynamic and integrated models. A little bit of scenario as well here, and a little bit about a different environmental scenario dynamics in aquatic models. And lately I will talk about pesticides and how to improve pesticide big exposure calculation in mountains. So it's a peculiar situation where a few things happen in a different way. So what are environmental fate models? We know that models and fate models are predictive tools. So we use them to understand and try to figure out how chemical will spread and distribute in the environment. But of course, they're used in many other applications as idea for research. Sometimes we use models to try to understand why things happen in a certain way. And this is done also for fate models. Or, as we know, for regulation, we need to produce PEC or PACs for risk assessment in order to evaluate if exposure concentration are too high or nearly too high to the predicted no-effect concentration, like NEC, for example. Sometimes models are used in monitoring because models are mass balance object and capable to understand to explain why things happen in a certain way. And for example, we can figure out if monitoring data are wrong or are not properly organized, and maybe we should organize them in a different way. I'll give you a very simple, example of a simple model. It was an old model of ours, SoilFug. It was a very simple model, like basically a box model with one box with soil, but it gives an idea of how many fate models, the multimedia models we are working on, are made of. It's based on a well-mixed compartment idea. So for example, soil is well-mixed. This means that if we calculate concentration in soil, this soil will have one concentration in the box for each, for example, day or hour or whatever we use for calculation. It's a fugacity-based model, which is a peculiar way of calculating concentration based on Don Mackay's approach. And this model was very simple and is still used to predict concentration in soil and ditch water. So because it incorporates a number of fluxes, to the contribution of rainfall to movement of chemical and water in soil, volatilization, degradation, chemical emission, and then runoff towards a ditch, for example. We generally started in when looking at modeling, fate modeling history from very simple fate models, such as the unit world model by Don Mackay, which was developed in the early 80s, basically. So there was a simple steady state model, as many other models, and then we figure out that some dynamics, especially temporal dynamics, would have been important. So some of the model turned to be more dynamic in terms of unsteady stateness. And this unsteady stateness means dynamic in time. So we have initial concentration, for example, discharge of pesticides, and this concentration may, for example, decrease in time. But we also realize that other dynamic is important. For example, spatial dynamics. In fate models, we have one box system, and this one box system, maybe one concentration was not enough. So we needed to define more spatial dynamics. And very often to incorporate spatial dynamics and temporal dynamics to have a better picture and a better description in predictive term of the environment. Is this all? Is this what we need? What is missing? In the model developed, the first model developed, the environmental scenarios, which I mean by how the structure of the environment is described, was static, did not change with time. So even if the models were dynamic in terms of chemical emission, were steady state in terms of environmental characteristics. So the environment was not changing with time. But we know from ecology, from many other disciplines, for microbiology, from zoology, and so on, then the environmental scenarios do change in time when they are trying to describe real scenarios. Why is that? Because they have to incorporate physical and chemical changes, for example, temperature, wind speed, rainfall, and rainfall change in time, chemical degradation, for example, but also biological changes, for example, biological degradation or life cycle of organism to account for their exposure in time. So we needed to incorporate more scenario dynamics in unsteady state models. So I'll start with some terrestrial and aquatic dynamic integrated model examples. And I will start with the first model we developed with dynamic of air compartment, which is called AirFug model. And in order to do so, I have to tell you a few things about the planetary boundary layer. I'm sure most of you are familiar with that, but just to briefly explain, we mean, and we use the definition of Stoll. Planetary boundary layer is the part of the troposphere which is directly influenced by the presence on the Earth's surface and responds to the surface forcing within a time scale about an hour or less, which means the part of the troposphere, the part of the atmosphere, which is very much influenced by the contact or the dependence from soil, for example, soil temperature. And why is this important? Because planetary boundary layer it's a very dynamic compartment. It's a compartment which changes, for example, height during the day. If we look at the typical day, sunny day, starting with sunrise and going to noon and sunset and night, well, we see that the planetary boundary layer will change in time. And this height of the atmosphere will go from nearly a few hundred meters, maybe 100 meters at night, to maybe 1 kilometer or even more in summer, 2 or 3 kilometers. And why is that? Because the sun will heat the surface of the soil, and the soil will release heat to the surface atmosphere, to the surface layer, to the bottom layer of the atmosphere, and creating some convective layers of air, bubbles of air moving up, and cold air moving down. This warm and cold air will create these convective currents and will mix the whole planetary boundary layer. So the mixing layer in which pollutants are diluted will change very much from sunrise until noon, and then will drop significantly at sunset. And a so-called stable boundary layer will be formed at night. So the pollutant emitted at surface level, at soil level, will be diluted in a very narrow layer. So mixing height of probably at about 100 meters. So we try to describe this, and we defined the so-called AirFug models, which is a dynamic, simple three-box model: upper air, lower air, and soil, where the lower air was a compartment moving with height. So every hour the height of lower air will change according to PBL layer. And the lower air and PBL will change with temperature and also for wind speed, with wind speed. This model was developed a few years back and was used to predict the concentration of PCBs in air, measured in Zurich by Gazic et al. Also, Matthew MacLeod was authoring this paper. And they measured PCB 52 and 149 in air at 4-hour interval. And these are these yellow triangles. And we, using PBL change and AirFug, we model concentration in air at 4 hours interval or continuously, and we were able to predict, I would say in a nice way, the behavior of PCB concentration during day and night. And then we developed an additional model which is called AirPlus. Why plus? Because it started with AirFug and incorporated an additional model, an existing model called AirMod, and a US EPA model, it's a physical-based model, which basically calculates concentration in air on a simple Gaussian equation approach. And this model will based on emission, on an emission rate, for example, combustion of some products, for example, emitting PAH in the atmosphere, and will produce concentration changes in air, and this concentration changes will produce input for upper air and lower air, and this AirPlus integration will be able to process in a multimedia fashion the concentration in air. If we look at the bird's eye view, so for example, from top, we see wind direction in the 4 cardinal points, and we put the emission, which was a stack in the center of this system, and we put the AirFug box, which is a small domain, 100 by 100 domain within a larger domain of 10 by 10 kilometers. We can see how the air concentration distributed by this model, depending on wind direction, wind intensity, and so on, will affect the multimedia box calculation. So what we did is we calculated, and this is a representation of 2 typical days, one winter day in December, one summer day, where the mixing height or PBL height is very, very different. In winter, at night, you see it's very, very low, and then during the day it raises up to 500 meters and then goes down to maybe 100 or so, while in summer at sunset, at sunrise, it goes up to 1,000, 2,000, even more meters, and then drops to 100 meters at night. So while the mixing height is very different, so is the concentration in winter and summer, depending on this emission. While the concentration doesn't change very much because of internal emission in the AirFug, when the contribution of the point source is arriving to the box, it is capable of raising concentration up to a factor of 10. So one order of magnitude in winter, a little bit less in summer, and this is due to the huge dilution. So this is an example of how the scenario of the winter and summer scenario, two typical days, could be different in terms of exposure and air concentration. But this also has the effect of soil concentration, because AirPlus model can calculate the deposition of chemical to soil. In this case, we have phenanthrene concentration. During 7 years, you see that this line is actually the multimedia phase model calculation, and the dotted line is the multimedia box calculation with an addition of a point source contribution. So you see that even soil receives chemical in addition to that, which come from the single point. And this can be seen also in terms of soil concentration or deposition flux with some, for example, in a year simulation, there are times at which deposition may reach higher levels, and this is important and raise the overall concentration in soil. So the integration of a physical-based model with a multimedia-based model can use both advantages of both models in calculating concentration in air and in soil. The model was then upgraded to a newer one called SoilPlus, where the single soil layer was modified into a multi-layered environment. So we have many layers of soil and many layers of litter. And we could discharge chemical in lower air as well as litter or soil. So this chemical is capable of running many mass balances, so for chemical, water, and organic matter. So we have organic matter change in time as well as water change in time. And we ran this model for hexachlorobenzene, which is a hydrophobic, semi-volatile chemical. And we evaluated the amount of deposition, basically, of hexachlorobenzene to soil, basing on the presence of soil only, and this is the simulation on the bottom, this thick line, or the presence of two different types of litter. So the intermediate line, this dotted line, is the degradable litter, so a litter made of organic carbon, which is not very persistent, while a persistent organic matter, organic layer, litter layer, will increase deposition to soil. So the importance of a multimedia fed model is, and the importance of the dynamic of the scenario is reflected, for example, in increased concentration of soil. And this is also true, for example, for different addition of organic matter to soil, such as biosolids or amendments. This model was later modified by including a vegetation model, so SoilPlus structure with a 3-compartment foliage stem roots model, which was later published, and is a fully fledged multimedia fed model which can be used to understand the fate of chemicals, and then will be later used in a later example. In terms of aquatic example, I will show you briefly the EcoDyna model, which is a model which was due to the incorporation of our existing Dyna model, a basic water sediment dynamic model, and a new single organism model, which is a model where a single organism, like a fish, can change in time, where volume, growth rate, lipid fraction, and feeding rate can vary with time. So, for example, we can follow stages with time of the fish growth. And these are some examples of changes in time. We have some measured data for female and male corygonids. And so fish weight was changing in time, and so was lipid fraction and feeding rate. So we have like the winter period and summer period and so on and so on. So all this dynamic organism behavior are very important when we have to run a simulation. And we ran the simulation of a persistent chemical, which is DDT, DDT, which was discharged, unfortunately, in Lake Maggiore in northern Italy. in the 90s, and the model without simulation of food uptake predicted not very high concentration, but when we incorporated feeding rate and all the parametric, as I explained before, we had a very good fit of concentration in time. The model was later modified, and this was a collaboration with Paul van den Brink in the ChimERA project and other colleagues like Frederick de Lender and other colleagues. And we modified the dyna model to give more dynamicity and to add more compartment, for example, a layer sediment compartment, a microphyte, detritus, dissolved organic matter, DLC, and so on. And so this is a way more dynamic model where many biological and non-biological compartments can vary in time. We also have a dynamicity in terms of space. For example, we also have slices of water where the chemical can move, can be discharged in the initial canal and then move through the pond. For example, it could be a pond, could be a river. And so we can follow how the dimension of the slice and the characteristic of the slice, for example, dissolved organic matter or presence of macrophyte or other organisms could change concentration, or deposition to sediment could change bioavailable concentration. And there are some physical description of the friction, of the water flow, and so on, but I will skip this part. So this is an example of simulation for pyrene, which was initially discharged in the first box, which is a canal, part of a canal. So if we see the concentration in time where the macrophyte growth is present, we see concentration with a peak and a slower peak in time in the first box and the second peak later on. But while we follow the following slides, which are box 3, 4, and 5, as you see here with a different color, we see that the peaks in the boxes are still important, but look at the concentration here are 4 compared to 120 here, depending on the dilution, depending on the much larger dimension of the system, but also on the presence of microphyte, detritus, and dissolved organic matter. Okay, now I will move to environmental scenario dynamics, and which is another part of the ChimERA fed models, where we try to devise some ideas, some responses of temporal dynamics in different scenarios at European levels. So we try to develop 5 environmental or ecological scenarios, and we wanted to understand how the bioavailability of chemical would change in time in the different scenarios. So we have temporal profiles of water temperature, phytoplankton, biomass, detritus, and dissolved organic matter. We started from a lake, which is Lake Candia, which is in Piedmont in northern Italy, a very well-studied lake with a historical series of measurements. It's a meso-eutrophic shallow lake, and we used this lake to extrapolate behavior of many profiles, as I mentioned before, and we tried to apply this to 5 scenarios: a Scandinavian scenario, UK scenario, Central Europe, Northern Italy, which correspond to this one, and a Mediterranean scenario. So the results of this scenario were the development of equation to develop, for example, the phytoplankton growth, depending on temperature, depending on seasonal characteristics, seasonal length, for example, and so on. And this, for example, shows very, very different behavior, for example, with the red curve, which is a Mediterranean scenario, or the Scandinavian curve, which is the dark green curve here, in terms of phytoplankton biomass, and also in terms of detritus derived by phytoplankton biomass. And this different behavior correspond to different dissolved water concentration, for example, of chemicals of different hydrophobicity. So a medium hydrophobicity chemical, PCB 52, medium high, PCB 153, and highly hydrophobic, PCB 208, where the concentration change from the maximum to the lowest concentration will be very different depending on the chemical log KOW, so hydrophobic characteristics. And this can be translated also in this chart. This chart plot the POC, so the particulate organic carbon in milligram per liter against log KOW. And you see that the scale of colors correspond to a concentration of chemical, bioavailable concentration of chemical depletion. So the darker color, the largest depletion we have. So the larger depletion will be in Scandinavia, because of the smaller delta, and the lowest depletion will be in northern Italy. Third and last example will be about pesticide peak exposure calculation in cultivated mountain watersheds. Why pesticides? Because pesticides or plant protection products are chemical very much used in crops and for protecting crops. And we know that there is a lot of effort in developing peak exposure for this situation. And peak exposure could be very important because not very often we understand properly how chemicals are affecting organisms in the ecosystem, especially aquatic ecosystem. So why cultivated mountain basin? Because we have situation in which runoff water moves very fast to water bodies, much faster than in more plain environments. And peaks of chemical moving to water could be shorter in time, so much shorter but larger, higher, much larger in height. What we did was developing two approaches, one called DynaPlus model, using SoilPlus, and DynaNet, which is a modified version of Dyna model of river section. So there are many sections of Dyna model in rivers. So we have a soil unit in which we have application of chemical. We also have spray drift calculation, and we have runoff of chemical to the water model. And this is spatially resolved, so GIS based, and could calculate concentration in runoff water, also in steep situation where the slope is important. So we applied this to so-called Novella Basin in northern Italy, in Trentino Alto Adige region. So coupling SoilPlus model and DynaNet with a few physical chemical properties, air compartment, soil properties, and so on. And also the DynaNet model was using some physical property, for example, the geometry of the links of the canals, the suspended sediment properties, organic carbon fraction, and so on and so forth. And this allowed us to calculate concentration in water. We also improved the DynaPlus model with the SoilPlus bag model, so the model which included vegetal biomass, with a ChimERA fade model in order to have a better description overall, for example, macrophytes and dissolved organic matter. So we have both terrestrial vegetation and dissolved organic matter and primary producers in water. So let's see the first case. The first model integration was run on the Novella River, about watershed about 133 square kilometers, 7 links, as you can see here in the picture. And we have apple orchard, about 1,400 hectares. And this orchard are in this situation, so very, very, very steep situation sometimes. So you have situation in which slopes can be up to 52% of, on the average, 22.5%. We coupled this simulation to a measurement performed by some colleagues, in which chemical was collected and measured at the outlet of the basin, which is the red point right here to the left. And the model was parameterized for year 2012, and results will be shown for chlorpyrifos. The second case study is the one developed for the Adda River. Adda River is a river which starts in the Alps and goes through Valtellina, which is a valley, a very east-west valley, very close to the Switzerland. Also, in part of this valley, there is a large orchard, apple orchard area. And so another situation where we could compare the discharge of chemicals to a river. And this is the picture of the lower part of the area. So you see the slopes with the orchard and the Adda River here. Okay, so this is the first case. This is a Novella River. And in the year 2000, you see concentration in water. And this is actually spray drift. So the concentration here is not due to runoff, but the peaks of runoff are this blue peaks on the right, and the green dots, the green squares, are actually the chemical measured concentration. So we can see that the model predicts pretty well some concentration measured. In the Adda River, we ran the simulation with two different chemicals, chlorpyrifos, the same as before, which has an intermediate log KW, so hydrophobic chemical, but not too much compared to the other chemical, etofenprox, which has a log KW 7.05, so much more hydrophobic. So we would expect, which would stick to suspended sediment and sediment more than chlorpyrifos. So again, we have drift, and we have peaks here, you see, which were compared to some toxicological value. This is Daphnia EC50, so acute toxicity value. We can see that for chlorpyrifos, although these peaks are very short in time, maybe a few hours, we are very, very close to the range of toxicity, very, very close to acute toxicity for chlorpyrifos. While for etofenprox, which is way more hydrophobic, so concentration are typically much lower, the runoff peaks are still important, but way far from the toxicological acute toxicity values. Just one of a few left slides. If we compare the loss processes, we see that chlorpyrifos losses are mostly depending by water outflow, so chemical will move through water moving out of the system, while etofenprox will be probably depending on the suspended solid outflow, so suspended solid moving with water. And not really dissolve water concentration. So to the conclusion, and I hope to leave some time for discussion. Dynamic scenarios are important for many situations when PBL variation is important, when vegetation is important, when POC and DOC is important to surface water transport of chemical, but also in vertical groundwater. We recently have some data of vertical movement of even very hydrophobic chemical with DOC and POC, small particles moving in soil through the pores of soils. And they can be very relevant in certain situations, for example, when the bioavailable concentration is important. And of course, you know the bioavailable concentration is the concentration responsible for toxicity. And of course, when we talk about this model and the model we've seen, we of course need more data and calibration, validation exercises to be sure that we could do a good job. And of course, most of these models, the model I've shown so far, are working nicely for apolar chemical, for standard, so-called standard chemicals, but not very well suited for polar or ionized chemicals. So a strong effort should be put in modifying this model to be capable for running polar or ionized chemicals. And with this, I thank you very much for your attention and will be open for questions.
Great, thank you very much, Antonio. That was a really, really interesting presentation, and I'm really looking forward to discussing all this with you more within our work package over the next few days. Are there any questions from the audience at this point? And again, you can either raise your hand or put it in the chat box, or I guess to start off with, just unmute yourself and ask. I do have a question to get us going. Antonio, at each time step within a lot of these models, especially ones that have really short time step on the scale of hours, like you were talking about, is the assumption of equilibrium made between phases or media? And if so, would there ever be a concern that if you shorten the time step enough, you're not going to reach equilibrium between phases, like for a particular chemical? And if so, I guess, how would you address that maybe?
Well, yeah, in terms of equilibrium, I mean, and you mean in terms of partition coefficient. I mean, partition coefficient do regulate behavior, and in this type of models, as well as mass transfer coefficient, which are kinetic limitation to this equilibrium. So there is a partial limitation of that. But of course, we are aware that for certain chemicals, but mostly hydrophobic chemical, equilibrium might not be such a rapid process. And so in this situation, maybe we will need more time to reach a truly equilibrium situation. Yeah. Mm-hmm. The time step in the model is actually, there are different time steps. Like there's a time step for displaying results, which is usually in terms of hours. Mm-hmm. But the time step of calculation is very often, and especially with these very, very dynamic models where there can be very rapid changes in, for example, meteorological condition, or for example, PBL can change of 100 meters 1 hour to another. So in that case, the calculation can be even at seconds level. Mm-hmm. It would be very sharp change. So yes, so we must be careful in that circumstances.
Any other questions? Annika, go ahead.
Thank you. Thank you, Antonio, for your presentation. Very interesting. Yeah, I have a question. Since you mentioned in the end that there's a big lack of information to actually model the ionizable compounds, I just wanted to have your insight on whether where you see the most difficult question to answer there. Like, is it just because the physics is more complicated, or is there less data, or can you give us an insight on that?
Yeah, well, most of the models were born with the concept of hydrophobicity, so log KW, so a very, very, very simple partition coefficient, which was used to measure the hydrophobic property of the chemical. And this log KW was used to predict some partition coefficients, such as the organic carbon, water partition coefficient, and then soil water and sediment water partition coefficient. And being able of having predictive equation like that was the start of this mass balance model. But now we don't have, or better, we do have just a limited set of equations capable of predicting the behavior of polar and ionized chemicals, especially when they change species. for example, during a pH change, for example, depending on the APKa, for example. So we need data to predict the equation for these chemicals, and we need also data on their behavior in the environment. So when we will be able to have models like this, and we will be capable to produce results, we will also have to have data for these chemicals. Unfortunately, most of the rich chemicals, and the large number of rich chemicals are like those, so are dissociated in polar chemicals. We are desperately in need for new measurements.
So you're saying that's also not that easy to detect, like when you take a water sample and you want to measure these compounds?
You didn't know well, that's a part of the reason. Now we have LCMS, so we have the capability of measuring with very sensitive instruments, also polar ionized chemicals in different conditions, and also the fact that we have GC, for example, GC and GCMS in the past as a tool for measuring concentration, for analyzing, for measuring concentration of the chemical. But GC, of course, was mostly devoted to volatile chemicals, so apolar chemical. So people are starting to find out more and more polar chemical, also because we have new analytical tools. But we need more than polar chemical measurement. We need also to evaluate their behavior in the environment. So we need to change our attitude and structure also.
All right, thank you very much.
You're very welcome.
All right, we still have time, about 4 or so minutes for more questions. So if anybody has any more, feel free to include them in the chat box or raise your hand or unmute yourself. Okay, so does not look like there are any more questions at this point. So thank you again very much, Antonio.